Our next speaker is Sara Rouhanifard, who is an assistant professor at Northeastern University in the Department of Biomedical Engineering bioengineering. She's bringing some medical to it though. And she's going to talk to us about nanopore sequencing of RNA. Sara, take it away.
So yeah, I'm an assistant professor in the bioengineering department at Northeastern. My lab is primarily interested in developing tools to image mRNA modifications. And when I first started the lab, what we realized was in order to do this, you really need to have accurate maps and quantification of the mRNA modifications. And this led us down a path to do direct RNA sequencing to map the Epitranscriptome so that we could develop this other tool. So I'll talk to you about that today. So I'll just jump right in and tell you about how RNA has over 150 different chemical modifications beyond the canonical A, G, C, and U, with the fewest found on cytidine and the most found on uridine. So these modifications are found on all classes of RNA, with the most diversity occurring on the tRNAs, but also a fair number on the ribosomal RNAs and a growing number of them found on non-coding and messenger RNAs. The modifications we find on the messenger RNAs we term the epitranscriptome, which is what I'll be talking about today. So tRNAs are also the RNA type that has the highest percentage of modification. They're also really well characterized due to the high abundance inside of cells. So by mass, our ribosomal RNA occupies close to 90% of the total RNA in mammalian cells. We have less tRNA by mass, but by molar levels, they're higher than that of the ribosomal RNA. mRNAs, on the other hand, are 1 to 2 orders of magnitude lower than the ribosomal RNA, as well as the tRNA. The reason I'm telling you this is that the abundance of the tRNA and the ribosomal RNA made them amenable to mass spec techniques, so LCMS. So we know a lot more about the modifications on these molecules than we do on non-coding RNAs and mRNAs. For example, the ribosomal RNA can be digested by nucleases down to individual nucleotides and then subjected to LCMS. So what you're seeing on the top right of the screen here is 50 ribonucleoside standards that have been run on LCMS. So each of these numbers is representing an individual ribonucleoside, and then the sample is run below. So based on the retention time and the mass, you can identify which species you're looking at from your ribosomal RNA. So when you compare to the standards, you can get the precise quantification because we have such a large amount of them inside of our cells. Now, mRNAs, on the other hand, are just 3 to 7% of our total RNA by mass, but they are substrates for modification. So our landscape for mRNA modifications is incomplete, both with incomplete maps of known modifications, and you can see the known modifications on the screen now, but they're also yet to be discovered mRNA modifications. So current literature, we're finding more and more mRNA modifications being discovered. So we're going to need new technologies to fully characterize the epitranscriptome. So even based on the limited information that we know that individual mRNA modifications, and what I mean by that is one gene that has a single modification at a single site. We know that even those one sites of modification can have extreme physiological consequences. A really classic example is the adenosine to inosine RNA editing that occurs on the glutamate receptor 2 in neurons. So in neurons, I find this modification really interesting because in neurons, this modification needs to occur close to 100% of the time. So I'm showing you a wild type healthy neuron that the modification has to occur in. And the modification is preventing calcium influx into the neuron. In an unmodified or unhealthy neuron, calcium can enter the neuron, which leads to severe cognitive impairment. So another really interesting example is the M6A modification, and this has been shown to have both a writer and an eraser, demonstrating that it's reversible, and drawing a really interesting parallel with DNA modifications that we know a lot more about that are known to regulate gene expression. So we have some solid examples of RNA modifications having interesting biological functions, but we don't really understand what they're doing inside of our cells at specific sites. So a major hurdle at the moment is that they're really challenging to detect. So the adenosine to inosine RNA editing is perhaps the easiest one to detect. And people who study this probably wouldn't like that I called it an easy one to detect, but it's easier because inosine is, it results in a change in the Watson-Crick base pairing. So A pairs with T, I will pair with C. So based on that, you can compare the cDNA to your genomic DNA to identify sites of putative A to I editing. Likewise, M6A is also amenable to sequencing thanks to the development of an M6A specific antibody. Other modifications, such as pseudouridine, are not so lucky. These are a little bit more challenging to detect because they don't change the Watson-Crick base pairing. Uridine pairs with A the same way that pseudouridine pairs with A. There's no antibody available, and they're not really amenable to mass spec because they're mass silent. So this is true for a lot of RNA modifications, and this is where part of the challenge lies. So some of the, just to give you a really brief overview of some alternative methods to detect mRNA modifications, you could use LCMS as I described in a previous slide, which is destructive. This is breaking down your nucleotides. You can do this in a non-destructive manner, but that's very low throughput and it requires a lot of input. There's also a method called Scarlet, which I won't get into the details of, but this is a site-specific cleavage near the site of a modification and radio labeling of the modified sites followed by TLC, so thin layer chromatography. Also a low throughput method, but it's something that's very reliable in the field. You could also use antibodies in high throughput sequencing. So I mentioned the M6A antibody, which is used to do Illumina sequencing with and identify the M6A sites. Something that's very common is activated nucleotides in high throughput sequencing. So this is using some kind of chemical labeling method to identify the modification site. For example, with pseudouridine, you could use a chemical called CMC, which will site specifically label it. And then when you generate your cDNA library, you'll have an RT stop at the site of the modification, which helps you identify where they are in the sequence. PacBio also has a method to detect M6A residues. So in this method, really briefly, they enzymatically introduce a C to U deamination at sites adjacent to the M6A residues. And then you can detect it by cDNA sequencing. And then finally, we have the Oxford Nanopore direct RNA sequencing, which I'll talk about now for the rest of the talk. So with the Oxford Nanopore platform, which Mark just really nicely introduced. So I'll just really briefly talk about this image again. So briefly, you have a helicase motor that passes the RNA through the protein pore using an applied voltage. So as the strand advances through the pore, you can see single nucleotide steps. The ionic current blockage is reported as a function of time. So this ionic current signal is then converted into your RNA sequence, so A, G, C, and U, using a machine learning algorithm that's trained on known RNA molecules. So in order to identify sites of RNA modification, it's important to include controls that show deviations in the signal that will tell you where to look for the RNA modification, because the goal is to identify sites de novo. So I'll show you several examples of how to do this to try to give you an overview of how the field is thinking about this in different ways. This is not an exhaustive overview because it's just a 20-minute talk, but I'll try to hit on the ones that I find to be the most different and why. So one way, which I'm showing you on the screen, is to generate a matching unmodified transcriptome. So for direct RNA sequencing, you're taking your native mRNA and you're generating a library, which you'll pass through the pore directly. If you want to create an identical control to this, you can make a matching unmodified transcriptome by reverse transcribing your entire mRNA library from the same cells and to make cDNA, and then in vitro transcribing it using canonical RNAs. And you've basically made an erased transcriptome. So you removed all of the RNA modifications from these samples, and then you can pass this through the pore and collect your direct RNA sequencing data for that transcribed mRNA control. And this has been used by several groups in order to identify sites where you have a deviation in the current signal. So one such control was used to explore the current signal distribution at the site of a known M6A modification. And they found that there was an ionic current signal deviation caused by the M6A residue. So you can see that your canonical 5-mer or K-mer sequence is shown in orange. And you can see in the presence of the M6A, you're deviating from the normal signal. So you're deviating from the normal current blockage that you're seeing. So this result was validated using a synthetic construct. So this synthetic construct is an oligomer containing a modification ligated to a carrier RNA and then passed through the pore again. So again, here you can see that your canonical nucleotides, your 5-mer sequence, your K-mer sequence is running at about 122 current. But when you introduce the M6A modification in the center, you're deviating from the current signal. So soon after that result, another group reported a systematic base calling error at the site of M6A. So in the previous one, they're detecting the M6A based on a deviation in the current signal for that 5-mer sequence. So this group is reporting a systematic base calling error at the site of M6A. And this was determined using synthetic constructs that this group calls curl cake constructs. So this is oligomers containing all possible 5-mers and introducing an M6A into every single site where an adenosine would be incorporated. So you have a lot of M6As introduced, but you're covering a lot of different types of 5-mers on here. So, and this is an example of what these constructs would look like on the top right. So your M6A modified, you can see that there are a lot of errors introduced here, and these are base calling errors because you're introducing a non-canonical nucleotide, your M6A. And then in the unmodified version, you have some errors that are showing up, and these would be standard K-mer errors. So whatever is an error in the M6A modified that's not an error in the unmodified is a difference caused by the modification. And you can see that when you zoom in right here. So these controls have the benefit of covering all K-mers. However, the downside is that the modifications are really close to each other, and this interferes with the signal at the pore, which we'll discuss as I progress through the talk. So what they did was they extracted mRNA and sequenced for yeast using wild type and IME4 knockout yeast strains that lack M6A. So the green asterisk that you're seeing on the right is highlighting the M6A modified nucleotide in the wild type and not in the knockout strains. So you can see that this base calling error faithfully shows you where the M6A modification is, and it's not showing up in your knockout strain. So first we distinguished M6A using current deviations and then distinguished using a base calling error. So this base calling error was a particularly strong feature for the pseudouridine modification. And I'm showing you an example of work from my lab, but this was done by several labs and also in ribosomal RNA, not just mRNA, but we weren't the only ones to report this base calling error. But just to demonstrate what it looks like, if you look at sites from the human transcriptome that we know to have a pseudouridine modification, that site is in the center of these 5-mer sequences that I'm showing you here. If you compare your direct RNA sequencing to your IVT control, which is your unmodified transcriptome, you can see that you have a systematic U to C base calling error that's showing up at the site of modification, and it does not show up in your IVT control. And then we applied this to the transcriptome, and we overlaid it with other methods using Illumina sequencing to detect RNA, like pseudouridine modifications. And we found that it faithfully recapitulates the other sites that were picked up by these other methods. So it's an effective way using this base calling error to pick up where the sites of pseudouridine modification are. The problem with this is that this method is not quantitative, and the error varies based on the signal sequence of the particular k-mer. So it depends on what this sequence is. And it also varies based on the sequences surrounding the k-mer. So several groups have attempted to correct this error using machine learning algorithms and different training sets. And I'll highlight a few examples in the next slide. Again, this is not an exhaustive list. So in one instance, the algorithm was trained on a wild type and knockout strain of yeast with known size stoichiometries. And these stoichiometries were determined by LC-MS. So the algorithm extracted and trained on 4 signals features in this case. And these signal features were used to determine the absolute stoichiometry, and they found that the stoichiometry was similar to that which was reported by LC-MS. This algorithm is called nano-RMS. So shortly after, to expand on the coverage of modified sites, because that was limited to just the enzyme that was knocked out. That was called a PUS1 knockout, which is an enzyme that produces pseudouridine, but it's only 1 of 12 that produces it. So to expand on the coverage of sites and possible k-mers, another group extracted ribosomal RNA from 5 different organisms and pooled them and subjected these samples to RNA bisulfite sequencing to identify sites of psi modification and quantify them. So this provided their training data from which they extracted 12 signal features. So in the previous one, it was 4 signal features from a knockout strain. Here it was ribosomal RNA that has a lot of modifications. They identified the pseudouridines, quantified them, and extracted 12 signal features that were found to be the most important. So this algorithm is called nano-SU. So we have nano-RMS in the previous one, and then nano-SU in this one. So another example to maintain the long-range sequence context rather than just the 5-mer sequence context, our group generated 1,000-mer synthetic controls that matched the mRNA sequence that bears the pseudouridine in the center of them. So we generated 100% psi and 0% psi controls. So in the 100% psi, I mentioned this earlier, the base calling error is not quantitative. So if it was quantitative, you would expect that at the site of modification, you would have 100% C showing up, but we don't. So we need to correct for that. But since we have these very clean synthetic controls, the 100% psi versus the 100% U that were generated using solid phase synthesis, we can train with really clean training sets. So we train a machine learning model on the 100% psi and 0% psi synthetic controls and corrected for this, bringing our 40% mismatch error, which was detected by U to C base calling, and we brought it back up to close to 100% as expected. Then we applied this algorithm to the human transcriptome, and we are able to apply for those specific sites that we have the synthetic controls and find out the specific occupancy. So an interesting thing to note, most mRNAs have very low coverage, and we showed that using this algorithm, so they're usually in the 0 to 25 copy regime when you're looking at nanopore sequencing. This presents a sampling challenge. So using our algorithm, we could accurately predict the psi occupancy in even low abundant mRNAs, which is really important to consider when you're thinking about how to quantify your occupancy for mRNA. So just to give you kind of a summary, and as I mentioned a few times, this is not an exhaustive list of what everyone is doing. There's so many people working on this, but there's a lot of different types of controls. And these are really interesting for us to explore all of them, because none of them are perfect. And I'll tell you really briefly why they're not perfect. So you can have the synthetic control that is very long and covers every single k-mer. The problem with doing this is that if you're in vitro transcribing every single k-mer and you're just replacing replacing your nucleotide with a canonical nucleotide, you have a lot of modified sites that are in close proximity to each other. And when they're too close to each other, you're looking at an image of the side of how an RNA would pass through the motor and the pore. So if you're just looking at a 5-mer sequence that's passing through the pore, it's going to be influenced by longer range sequence contacts. And the field generally thinks this is going to be about 11 or 12 nucleotides that are going to interfere with the signal at your site of modification. But this is kind of unknown at the moment. So the synthetic constructs we like because they could cover a whole bunch of things and it's cost effective. But the problem is you have to replace every nucleotide. So every U has to be replaced with a pseudo U, or every A has to be replaced with an M6A. And you have this problem that a 5-mer sequence or an 11-mer sequence might have multiple U's in it, and that could interfere with your signal. Also, there's enzymatic knockouts. The problem with these is that we don't understand the enzymes well enough to be able to cover this transcriptome wide. If you knock out one of them, is another enzyme going to come in and take over for it? If you knock out one of them, are you going to have pleiotropic effects across your cell? We just don't know. And also, like, the enzymes might only cover 20 sites. If you knock out a single enzyme, you might only be able to quantify 20 sites. Native ribosomal RNA is a control that's been explored. This is an interesting control because it's native. You're looking at natural RNA sequences. However, ribosomal RNAs also have a lot of different types of modifications that are very close to each other. So you still have that hypermodification problem. Synthetic mRNAs are kind of interesting. But they're extremely expensive. Like, generating one of those 1,000-mer sequences that I described to you is $800 because synthetic RNAs are difficult to synthesize, and making long ones is impossible to synthesize without using a ligation strategy. Also, you're making an assumption, or we made an assumption, that the mRNAs are only modified at one site and it doesn't have any neighboring modifications present. IVT controls that I described also are not perfect. These are nice to identify a nucleotide variant, but they can't differentiate between multiple uridine modifications, for example. So we've seen in our ribosomal RNA data that the U to C base calling error not only occurs at the site of pseudouridines, but it also occurs at methyluridines. So these are some of the challenges in the controls that the field is thinking about right now and trying to solve. So also challenges in developing the best training data. This is related to my previous slide. So how do we encounter the problem of throughput, and throughput means all of the k-mers, and how do you define what a modification looks like at every k-mer? And do we consider a k-mer 5 nucleotides, or do we consider a k-mer 12 nucleotides? There's a big difference in the scale of those two problems. The cost of RNA synthesis of standards, like what is the gold standard and how can we synthesize this in very high throughput? Satellite modifications are another problem. So if they're too close to each other, it causes a whole new error that is even more difficult to quantify. And it increases you need to increase your throughput a lot if you need to cover every single type of satellite modification that's possible. The length of the sequence context, I've mentioned this a couple times, like, are we interested in just the 5 nucleotide sequence, or do we need to expand to 12? Or do we need to go even further beyond that? How many nucleotides flanking the modification site are important? And then finally, controlling for same base modifications. So multiple different types of uridine modifications. We don't know which ones are on mRNA or where they are. How do we know we're detecting the correct one? So probably what's going to happen is we're going to need to attack this from all angles, which is what the field is doing right now. But these are just highlighting some of the really grand challenges for looking at modifications using direct RNA sequencing.
2 minutes, Sarah.
Okay, I'm actually done. I'm going to end there.
All right, I jumped the gun on that. Thanks very much. Feel free to call out some folks if you want to give some acknowledgements. That's fine.
Yeah, so we worked a lot. So my lab works a lot with the Wenunu lab at Northeastern, and we also work with Yaming Hao to synthesize our synthetic controls. But that field is enormous. A lot of people are working on pseudouridine. A lot of different labs are working on this at the same time. So